my turn is next, and I'd like to start by thanking the organizers for inviting me here. It's always a pleasure to come to Paris. And then I have a confession to make. I'm not a pain specialist. Nobody's perfect, right? What I do is study stress for a long time, and when we want to induce a stress effect, we go to Marshall DeVoe and we use his equipment. And we keep thinking this is a stress effect, and now we know better. So, in conferences, the speakers talk to each other, and I would like to refer to a lot of the questions that came up today by talking to you about micromanagement trade-off of pain diagnosis and treatment. And the focus of my studies has always been the signaling by acetylcholine in the nervous system. And the family of genes I'd like to introduce to you today are microRNAs. And I think that they can provide a lot of these answers we were talking about, because these are tiny genes, 100-fold smaller than regular genes. They don't produce any protein. What they do is they wrap around the target transcript and they block protein production. And they do that not only to one gene, but to a whole pathway. So what we get is a difference between men and mice, which is a real problem. Like all biologists, I like to play with mice. And I have clinicians in the family that say that I can only cure mice. But mice are not men. So we are talking about genes that talk to each other. They may share their targets. They may stop complete processes. And they might have gone through evolutionary changes. And there is a theory called the thrift gene theory that says that genes that helped us survived in our genome. And there might be genes that helped the ancient men to survive stressful or painful experiences. And when the ancient man was stressed, it was because a lion was running after him. He needed to have a faster heartbeat, a better reaction to the stressful experience. I get stressed when I see Donald Trump on television. But it activates the same processes. Okay, so we are talking about genes that might be different among us, and that talks to Marshall's presentation of single nucleotide polymorphisms. So there is a complexity, there is a diversity, and we believe that there is a pain-related impact. So we kept talking today about how pain affects the body, but the brain reacts to it. And our brain reacts to pain in different regions with different impacts. So there is context dependence also in the intensity of pain reactions. Yesterday I was working on this presentation, and then I got this message from Genetic Engineering and Biotechnology News, so freshly from yesterday evening. And they said chronic pain affects 50 million adults in the United States. Opioid-based pain relievers, which we discussed today, contribute to an opioid addiction epidemic in the States. They noted 19,000 death cases in 2014. And the use of needles, of course, led to a major AIDS outbreak. And so that in Indiana, there was a public health emergency last year because of overuse of painkillers. So, there's a famous saying that a scientist is someone who knows more and more about less and less. And I definitely belong to that group. And I'd like to compare our know-how to our know-how of the world geography. In 580 BC, the world had one living area, which was one continent. Everything was connected, and it was flat. In 1492, the world was already a globe, but no one knew that the Americas existed. It took over 2,000 years to teach us about the world we live in. What about the genome? I've been teaching Introduction to Molecular Biology for many years, and I've told many thousands of students that 2% of our genome encodes protein and the rest is junk DNA. But today we know it's not junk DNA. There are 1,500 microRNA genes. There are many other non-coding genes that we are just starting to understand. What we didn't know was not junk. It's really important, and we need to start to take it into consideration. So when Marshall says single nucleotide polymorphisms can change a protein, right? But they can also change beyond that, the interaction of a gene with the microRNA. And that would not make a change in a protein, but it could create a reaction that might even explain a placebo effect. So we talked about DNA, RNA, and protein, and the microRNAs block the transfer from RNA to protein. They are known about 30 years, but there's already a Nobel Prize for their discovery. And like 95% of the researchers work on microRNAs in cancer. We would like to know what they do in the brain and how they affect brain-to-body interactions. So this is my analogy for what microRNAs do. Can you see there are two little policemen in the center? Many cars, a lot of traffic, but the policemen are those who control it. So how do they do that? We can study that thanks to one more property of microRNAs that is very attractive. They are so small that we can synthesize an inverse sequence and obliterate them. So microRNAs are druggable entities. We can make an inverse sequence, inject it, kill the microRNA, and increase the amount of the target. There's already one microRNA-based drug like that in the market for people who don't react to statins. So druggable entity, a very important concept. Now here is how a microRNA does its job. The pink is the microRNA, the green is the target, wraps around it and stops it from working. What did the development of these genes do to our brain? So we know the human brain is the best. It's bigger, it's heavier, it's more complex. We can do more things than any other animal. And for decades, geneticists used to say, this is because there was an evolutionary jump, because regulatory genes changed, because massive changes happened. And they say, these are transcription factors. Well, we say, why is that? Because transcription factors bind short sequences, because they operate on a whole pathway, but microRNAs do the same. So we set up to study the question, could microRNA evolution affect human brain development? And when I say we looked at the human, we used the Human Genome Project. There is a website called the 1000 Genome Project, which has today over 2,500 human genomes from different countries, different ethnic origins, different locations. We can look at the Neanderthal men. By the way, do you know that the Carmel Mountain is the most southern place on Earth where Neanderthals and Homo sapiens lived together? So when I say that, people jump in the audience and say, well, you may be right, but the Neanderthal sequence was found in Italy, in Spain, in Russia. Yeah, that's true, but we are still the most southern site when the Neanderthals lived. We can use the DNA genome of many primates and other animals. And when I say we, it's one of those ELSC students who we were talking about, Shachar Barbash, today Dr. Barbash, working in the Rockefeller University. And what Shachar did was to ask, when did microRNA genes insert into our genome? How old are they? And what he could find was that in all vertebrates, there are very few microRNA genes. Most of them are newer than vertebrate development. If we look at the mammalian emergence, there are more microRNA genes. If we look at big primates, and we are big primates, many more microRNA genes, mainly functioning in the brain. So the very recent time of evolution saw a burst of development of microRNA genes that affect the way our brain functions, helps us to use tools, write poems, come to Paris to lecture. So there is an evolutionary aspect to this. So Bettina Nadorp in the lab asked, let's suppose signaling by acetylcholine is a process, it's a pathway. Let's suppose microRNAs control pathways. So there should be microRNAs that control more than one gene in this pathway. And she actually found 29 genes like that, of which 6 are primate specific. So if you wish, we as primates acquired better capacity to control the signals from brain to body. And when she looked at which diseases are associated, she found inflammation, which was mentioned already, anxiety, brain damage like stroke, cardiac disease, neurodegenerative diseases, and pain. So if that is the case, then let's go deeper into 2 of these microRNAs. They all have numbers, they don't have names. So 132 is a conserved microRNA, exists in all mammals. 608 is primate specific, and these are the 2 that we went deeper into. And our project is called cholinomers, microRNA regulators of cholinergic signals. So we talk about stress, and we copied the word stress from physics. The physicists would tell you that a stress is a power above which a body would break. And we use that in a psychological term. We know today that microRNA 132 goes up dramatically in the stressed brain. We know that this happens in the mouse, but also in humans with inflammatory bowel disease, which is painful and stressful, and the patients would tell you that if they are stressed, their pain symptoms would be worse. But now we want to look at those changes in our genome which do not modify proteins, but modify the interaction of microRNAs. So here is a microRNA that is primate specific, no mouse. Actually, we made a mouse, but we are still raising it. And this microRNA controls the hydrolysis of acetylcholine. And many people, a lot, carry a change that interferes with that interaction. So what will happen to them? They will have too much of the target because the microRNA doesn't work. But they will also have too much of the microRNA, which doesn't have one of its targets. So what it will do is to oversuppress other targets. And that would activate a domino-like effect. So we have one change of one nucleotide, no change in any protein, but a lot of consequences, of which Eric Kandel, one of my great heroes, would say the future of psychiatry is when it combines with genetics. So what we've done was to look at a cohort of 642 volunteers, young sports active volunteers, and we genotyped their gene. And we found that those that carry this change indeed have too much acetylcholinesterase. We also found that the change appears in African Americans up to 34%, also in the African tribes. But in Caucasians, it's 4%. In Israel, it's in between, 17%. So if you ever wanted a proof about population migration from Africa through the Middle East to the West, here it is. Carriers, one change, also had higher blood pressure. And they show 40% too much acetylcholinesterase in the brain, which is important because all Alzheimer's patients worldwide get acetylcholinesterase inhibitors for treatment per body weight. No one considers that some of them might need a higher dose. So we asked the following question. If these people are more stressed, they are more anxious, would that work for them, against them, or do nothing? And what we've done to ask this question was to take 69 soldiers who were volunteering to the study And Israeli soldiers are definitely under severe stress. These are mostly paramedics. Out of 69, 13 carried this change. And we've asked the question, and we now is Alon Simchovich, a student in the lab, and we knew that traumatized patients, post-traumatic patients, show hyperactivated amygdala in their brain and down-regulated frontal cortex. We did a brain mapping with Professor Talma Hendler, a psychiatric expert from Tel Aviv. We did find hyperactivated amygdala. But instead of showing suppressed frontal activity, these patients hyperactivated their frontal lobe, and they were not post-traumatic patients. Otherwise, they wouldn't be doing what they're doing in the army. So this leads to very big hopes. If that is the case, if they hyperactivated their brain to solve a genomic problem, could that serve as a treatment? Perhaps biofeedback could help alleviate stress symptoms also in other patients. So microRNAs are at the top of a pyramid of reactions to stress. Now, what do we know about the impact of acute stress expressed as fear in humans? You can find articles in the press. A person experienced a terrible earthquake, got a heart attack, and fell dead. Another experienced a tsunami, and so on. And the discussion of those papers keeps saying, we cannot study the impact of fear on whole populations because there are no populations that are routinely exposed to chronic fear. Well, let me doubt that assumption. Definitely, the Israeli population has been exposed to chronic fear for decades, and let's hope that this doesn't spread to other countries. So what we've done to ask that question was to look at the biomedical data sets of 18,000 volunteers who comes on an annual basis for the medical center for their annual medical test. Their employer pays for it. These are healthy people. And in Israel, and they all volunteer also to fill a psychological questionnaire. One of the questions, for example, is to what extent do you fear the impact of terror attack on your daily life? And we measure their pulse, the simplest measurement on earth. And what we find is that 740 of those, instead of showing the well-known slow decline in pulse with age, over the 4 years showed a consistent increase in pulse measures, which is a risk of cardiac attack. These are the same people who answer that they really fear terror attacks. These are the same people that showed inflammation. They had an increase of their pulse by about 10 beats per minute, and we don't know yet whether that reflects microRNA changes, but we know it reflects a change in their acetylcholine signals. So you would ask, why would stress affect things for such a long term? And we've done that test together with Eran Meshorer at the Hebrew University on mice. And what we find is that if you expose mice to an acute stress, again, borrowing the equipment from Marshall, so he would tell me, this is pain, it's not stress. They change the wrapping of the gene in their genome, so we see an epigenetic change. We actually offered the PNAS this cover, but they didn't like it. On the other hand, we got a very nice news and views. And you know what? Al Jazeera came to interview us about this study, but they didn't want to broadcast it. So last, why pain? One day I get a telephone call from this lady. Her name is Michaela Kress. She works in Innsbruck. And she created a pain research group, and she wanted someone who knows to do microRNAs. So I joined that group. Since then, we fully profiled the microRNAs in blood cells from all of the patients with different pain symptoms. And we use this way to decrease the microRNAs that are shared between the patients. And we are about halfway into learning about a marker and hopefully one day a druggable entity. So someone asked in this audience, what's the length of drug development? Unfortunately, too long and with very low prospects. But we need to bridge between mice and men and proceed the effort. So I told you stories, but the people who need to be thanked are the patients who donated their samples and their questionnaires. I told you that human brain microRNAs evolved recently, and stress modification in them may exacerbate both pain and aging-related disease, and cholinergic activity predicts the fear-related pulse increases. And I'd like to end by citing Virginia Woolf, who said that the vision of a novelist is both complex and specialized, and I believe this is true for researchers as well. Thank you for listening.